So Hatim, thank you so much for joining me here at the World Advanced Therapies and Regenerative Medicine Congress.
Thank you. It's a pleasure to be here.
Thank you so much. So could you start off by sort of introducing PL Biosciences?
Basically we are a biotech company, a startup company. The company was founded in January 2015 in Aachen. This is a small city close to Düsseldorf in Germany, and we come basically from the biotechnology research, stem cell research, and we founded the company as a spin-off of the university hospital. And we were focused from the day one of the company foundation to offer novel and new and innovative cell culture solutions that can facilitate the regenerative medicine research and field. And as I told you, we started like 2015. We changed a lot in our business vision and the mode how we can adapt those products to the market and so on. It's have been a very long way, but we are very happy that we took this decision 3.5 years ago.
Wonderful, I'm relieved to hear it. So thinking about sort of regenerative medicine and these advanced therapies, what are the unique needs that they have in terms of cell culture and media?
Safety. I would say safety is really like the biggest issue you have once you think about regenerative medicine and cell therapy in general. So in former times you may know that there was like some animal-derived cell culture solutions that have been used like over 50 years ago. And by those days, it was the safety issues were not really like what we can see that today nowadays. And people used to work with animal-derived materials without caring about the safety of the patients and so on. And I think for today, it's completely the picture looks completely different, because safety, as I told you, is one of the major issues that we have to talk to patient safety. And that's why this was one of the biggest challenges to change the concept or the way of thinking, moving from animal-derived components to animal-free components. And nowadays it's like when you talk with the regulators and when you talk to authorities and so on, they just have to get sure that whatever you are doing with the cells in patients, they have to be safe and secure.
Mm-hmm. So why isn't it safe or secure to use animal-derived cell culture?
Because I think there's two main challenges using animal-derived material in the clinical applications. One of those is animal blood-derived products will never be so extensive tested like human blood products. For example, when you go for blood donation, the blood will be extensively tested and screened against all known viruses like HIV, hepatitis, and so on. This is not done when blood derived from animals. For example, for fetal bovine serum and so on. This is one thing. And sorry, I lost. Yeah. The other thing is. Regarding the challenges, what we have is the transition of diseases came from animal. We know about this PSA scandal, what happened as well, and so it's a very high risk material that you never know how it ends, and that's why it's very risky to work with animal material, because it's not extensively tested, it's not screened as it should be, and there's a high risk of transition of animal diseases and so on to patients at the end.
Okay, so what are some of the alternatives?
Human alternatives would be the best at the moment. We need to know that we still really don't know what is is the factor that helps cells to grow, or particularly stem cells. So it's a sort of a black box that you do not really know what are the components of or ingredients of those sera or those cell culture media, and that's why you have either to use animal or human, and that's why we still have some fully defined media, but still doesn't work really as we expect, because we're still missing some or two components in the ingredients. And that's why if you don't think about fully defined, because we are not so far to know everything about the serum content, then we have to choose whether animal or human sources. And that's why getting a human material, first of all, it's more safe. Secondly, it's more natural, because it's more suitable to work with human cells in a human system and not in an animal system.
So if a researcher was thinking about sort of developing their own advanced therapies, at what point in that process should they be thinking about their cell culture and that process?
I think it's very important from the beginning to make some clear and serious thoughts about what kind of system you're going to work with, because once you start the process, it's a way difficult to change the process afterwards. So, and people think, okay, cell culture is not really the fundamental tool what I need in my process, because it's a way complicated. But at the end of the day, once you start with a product which is not scalable and it's not you cannot adapt it to in clinical application and whatever, then you have to start from the beginning. And that's why I always advise people thinking or planning for clinical trials or for whatever clinical applications, they do have really to think seriously about what kind of cell culture media they are using, because once you grow with your expand your production and you scale up your production, you will get through some new challenges, and this is something that is more important to have some thoughts at the beginning with.
And have you noticed sort of attitudes with researchers changing? Are they starting to think about this as early as they should do?
When I go back, let's say 3.5 years, when we start doing the business and talking to people about alternatives and so on, I think that the picture looks quite completely different from nowadays. It was like, okay, we heard about this and it sounds very interesting, but there still was not really that pressure that put people to think seriously about alternatives. Nowadays, the topic is getting more and more serious, because regulatory bodies and authorities putting this on focus, and as I mentioned, safety is one of the major aspects that have been taken serious nowadays, and the picture has changed a little bit now, and people get more aware that they need to change. It's not a matter of I can't think about this, but they know that it's now the day that they have to change and to switch to a more suitable solution or technique that is provide the safety for patients at the end.
So I think definitely as the industry starts to expand and people are treating more and more patients, I think scale up is definitely going to be something people should be thinking about. How can manufacturers make sure that the product that they're getting is consistent at these larger scales?
It depends. Of course, we are still working with a natural product, which is somehow limited and specific level. We still have to think from another way, because blood donation is something which is still life-saving. That means you will have to give blood donation for the next decades, I think. And the good point, what we are doing here, we are not collecting blood that has been produced or donated for our purposes. We are manufacturing, let's say, an outdated product, which means upon expiry, we collect the material and use it for our manufacturing processes. That means we are sort of recycling a material that anyway would be trashed in the bin. And taking this in considerations, plus that you have to have access of those blood platelets, what is our raw material. This makes it the shortage of such a supply not limiting at the moment. I don't know how it will look like in the next 10 or 20 years, but at the moment we are offering, or we are using a product which is in excess, because as I told you, it has to be produced anyway for any clinical, therapeutic or transfusion purposes, and we are not producing this in particularly for us for manufacturing processes.
Obviously, this may be one of your next answers, but what do you think are the remaining barriers in this field?
I think at that moment is the biggest challenge what we have is the regulation of such ancillary materials. It's a big topic, and once you come with a new innovative product, you have really it's like a very two sides of one coin, because on one side you need to innovate in new products, and you need to bring new ideas and to look for alternatives. On the other side, you need to regulate that as well to prevent any abuse of any of those new techniques. And this is like something you're pushing on gas, and the other one is putting on the braking or slowing the process. And regulators is good as long as you regulate in a way that it still helps you to push you to go forward. But if you want to control everything, I think it will make the things way more difficult to start to thinking innovative. And this is one of the biggest issues that we have nowadays, that we are dealing with something that the authorities has never been in contact with. So, for example, once we start the discussions with the authority bodies in former times about the product, it was very new, nobody knows about that, and they said, okay, we don't really know that much, but we don't have to put any regulation now. Now they start to get more to know the product more and more, and now they're starting to make some thoughts about this and to regulate it. So I think you need to find like a balance between pushing, supporting innovation and regulating the stuff. And from my point of view, I think this is the most difficulty that we have, that we over-regulating is not good, unregulation is also not good at the end.
And how do you think that could sort of evolve and change over the next few years?
Always looking for open dialogues as early as possible. This is also something that we always discuss and advise. To seek for the dialogue with the authorities even before starting planning for the process, or just to get them involved in the process and to find a solution, because once you work together, I think it's a way more flexible than coming too late, and then they have to put any kind of regulation, which maybe sometimes makes no sense. And that's why it's always better to have this a very open and very open discussion from as early as possible.
Absolutely. And what can we expect from PL Biosciences in the next few years? We see PL Bioscience as one of the main suppliers for platelet lysate, one of the best promising alternatives to the fetal bovine serum, and we are developing a lot of our products. So we are adapting to the needs of the customers. We are more adapting to the regulations and so on, and I still see more potential in this product only than having as a cell culture supplement, and that's why we are working working very hard on that, and it's really very interesting for us, because we see more potential in the platelet lysate as only having it like a cell culture supplement. And that's what we are working on. So I think one day we will have some therapeutic applications for the platelet lysate, which can help people also and patients. That sounds very exciting. So Hatim, thanks so much for joining me.
Thank you very much.